All right, folks, we're going to delve further into the topics of disinfection and antisepsis and sterilization, all of those means of controlling microbes outside of bodies, and which are done primarily to prevent infection from ever happening and or spoilage from ever happening. Now, I say outside of bodies, you can apply antisepsis on body surfaces, but these are things we wouldn't do internally. They're not going to work, say, to remove microbes from the gut or from any other compartment within the body, just the skin and mouth principally. Maybe a few other mucous membranes. So I left off last time just overviewing the vulnerabilities of organisms. So we got cell walls, cell membranes. We've got the processes of protein and nucleic acid synthesis. And then we have the proteins themselves, and we can attack those in various ways. So those are some vulnerabilities that are able to be exploited principally by physical and chemical methods. So we're going to talk about the physical methods first for control. And here they are. And I've already said a little bit about some of these, so it may seem in some cases a little redundant, but it bears repeating. So we're going to go ahead and start right here. So heat can be used, and it's classified as moist when we apply it together with water vapor usually, or it can be boiling liquid water or hot water. Or if you don't utilize any form of water vapor or water and apply just heat directly, that's dry heat. That can be implemented efficaciously as well. Cold temperatures will control organisms. Generally, they're not anywhere near as effective as heat. Desiccation is drying. All right, so you can dry things out. That can be effective because when you remove water, either from the environment or from within cells altogether, you dry them out. You don't provide the medium within which chemical reactions can take place. And that's really what the essence of life is chemical reactions. So if you stop those, you stop life. And then radiation, different forms of what we call EMR, electromagnetic radiation. That would be photons. Make this up. So photons make up light. Visible light is a form of EMR, but it's also present in other kinds of EMR, like ultraviolet radiation and X-ray radiation and gamma rays and so on. And those are effective at limiting microbes for sure. And then finally, filtration is listed here. This is more of a mechanical approach. I mean, it is physical, but this doesn't do any killing. This basically just keeps organisms from, or microbes of any kind, from going from one compartment to another because the pore size of the filter is too small. So air filters and filters for filtering liquids are utilized here, and they can be structured in such a way as to prevent microbes from slipping through. All right, so yeah, we got the moist heat versus dry heat difference. And so one interesting thing about them is moist heat's pretty effective, and it uses lower temperatures than dry heat. Okay, so dry heat, when you apply it, it has to be at a pretty high temperature generally in comparison to when we apply any sort of water vapor. And moist heat doesn't have to be applied as long usually. So 15, 20 minutes of inhibition, killing essentially, in the presence of moist heat is more effective, or maybe, it might be as effective or even more effective than applying dry heat for an hour or more. So the amount of time that you have to apply dry heat is longer. And the reason moist heat generally works so well is that it either coagulates or completely denatures proteins. Now, coagulation has to do with the fact that proteins, when they're in cytoplasm, the enzymes and some of the structural proteins, when they're in cytoplasm of a cell, they are in soluble form. They're dissolved in water, essentially, and they're floating around and doing their jobs. So let's say this is a cell. Here's its nucleoid. And these little dots are, soluble enzymes that are kind of floating around in there. If you apply heat, you'll drive off some water. So there'll be some water loss occurring. And some of these proteins will, instead of staying in soluble form and staying off by themselves, let me erase some of them here to show you what will happen next. You apply the heat. Let me just redraw it. You apply the heat. For one thing, the cell's going to kind of shrivel a little bit. It's lost some water, and these proteins start clumping up. And when they wad up like that, they form little clots, essentially. That's coagulation. And they don't do their job like they should otherwise, like they would otherwise. And then denaturation is going even further with that heating. And, if you have a protein that normally is a little glob like this and you heat it up too much, it changes shape. It loses its shape. And remember, with enzymes, shape is all important. That's going to determine whether the active site can be accessed by a substrate or not. So that's what moist heat does. And dry heat, which has to be applied for longer, it'll dry out cells, so it'll cause dehydration, which is problematic because there's no longer enough medium within which chemical reactions can occur. But and protein structures will alter. But ultimately, when you take the heating to an extreme extent, you catalyze the addition of oxygen to the organic material. All right, and what happens then? If it's hot enough, if the energy input's hot enough, you get burning, all right? You will literally cause generation of light and heat, and you'll wind up with ash, all right? So that's oxidation, essentially, extreme incendiary oxidation, and you'll produce ash. That'll kill organisms for sure. Now, the problem with that, though, is, let's say you've got something that is valuable to you, some instrument that you're using in a lab, and you want to remove microbes from it, but you want to keep that instrument so you can reuse it. Well, if the heat is so high that you're turning things to ash, and you have a relatively, delicate instrument, you're probably going to ruin it. So this is often not a good way to go. Moist heat application using boiling water, or if you want to go further with that, use an autoclave so you can sterilize. The autoclave, or use a pressure cooker. Then, hey, you'll probably be able to keep your valuable instrument intact. It's not always the case, but often it is. All right, so that's something to think about. What you want to do. If you've got something that's expendable, dry heat, burning it to ash is the way to go. If you don't really need it, it's expendable, and there's the possibility of bad microbes like, say, Bacillus anthracis or Mycobacterium tuberculosis, and there's a real high risk of being contaminated by that. Just burn it. Just burn it to ash. Okay, so that's one way to go. All right, so this compares dry heat application versus moist heat, how long it takes for them to work. You can see the temperatures for moist heat are lower than those used in application of dry heat, especially when we apply great intensity. And you can see, well, for point of comparison here, if you look at the temperature of 121 degrees centigrade using moist heat, you can see down here with dry heat, that same temperature in the absence of water vapor requires 600 minutes to be effective, to reach sterilization. With moist heat, 15 minutes. All right. So you tell me which one would be more efficient in a lab setting, right? So moist heat generally is the way to go, especially if you're working in a biomedical or clinical setting. Otherwise, you can opt for dry heat if it's appropriate. All right. So when we're talking about temperatures and the efficacy or efficiency, the value of these. Some different terms that can be assessed are the thermal death time versus the thermal death point. Okay, and so you got a specified temperature, some temperature, and you want to compare that against others with regard to how effective it is. So you pick a temperature and you apply it, and then you check to see how long it takes to kill organisms. That's the thermal death time. All right. So if you applied, let's say 100 degrees centigrade, you use that temperature to kill microbes, and it took 3 hours to reach sterilization. The question would be, well, if I elevated that temperature somewhat, what would the difference be, right? If I elevated it to 110 degrees centigrade, it might only take 45 minutes, right? So the amount of time that it takes for a given temperature to reach sterilization is the thermal death time. Let's kill all test microbes. That's the shortest time for that. And generally, as temperature goes up, TDT is going to go down, right? That's almost always the case. And then we have thermal death points, which are different, all right? Easy to, I guess, make the mistake of confusing the two. But so this is one temperature, all right, that will kill all of a particular organism in 10 minutes, all right? So it's the lowest temperature at which that can be achieved. And that's going to vary from microbe to microbe. This is microbe, specific. I don't know if I like that way to describe it. Yeah, but it's applicable to each microorganism, microbe in turn. So the SARS coronavirus 2 that we're dealing with right now, it's got a thermal death point. There is a temperature. Once we take it up to that level, that'll kill them all in 10 minutes, right? And E. coli has a TDP, and Staph aureus has a TDP, and so on and so forth. Right? So, there are those. And like I said, these are ways of evaluating the effectiveness of temperatures. All right, so this shows sort of a diagram of what you would normally find in an autoclave. They're much more streamlined these days and not quite as clunky and so forth, but this is kind of the classic design of an autoclave. Notice it's got this thick metal, this jacket all around here, and that's required because this is going to be used essentially to contain lots of pressure. So you got a lot of pressure in there. Water vapor is going to be injected into it. You don't want this thing to explode. So that's vital. And then you got this, looks like a bank vault. The lock on there can be cinched down tight, and it's not going to come open. Anyway, and there's got to be some water, placed in here somehow. This particular one is more high tech than some of them I've seen. It's got its own sort of steam supply, water's piped into it. Some of them you actually literally have to pour the water in there. But in all cases, if you apply this properly, you can achieve sterilization. And remember, that means killing everything. And so the standard is to turn this thing up so that it reads 15 psi. That's 15 pounds per square inch. And that's, higher than our own atmospheric pressure that we're dealing with normally. So 15 psi is usually where we want to set that at the very least. And that's going to let us get up to that temperature of 121 degrees centigrade, because without that pressure, the highest we could go is 100 degrees centigrade. So that pressure does that. And then we apply that for anywhere between 10 and 40 minutes. I think usually about 20 minutes is a good sort of happy medium, and that's what most people say would be their standard. But anywhere in that frame is usually adequate to kill everything, endospores and so forth. All right, so there's that. One of the stipulations is steam has to get to the surface of the item being sterilized. Now, if you want to recover it, if it's something that's very delicate, it's probably not going to make it through this process. So petri dishes with sterile agar and so forth. If you want to save a plastic petri dish, this isn't going to let you do that. It's going to melt. You had Pyrex, it'd be okay, right? It says must not be heat or moisture sensitive. That is if you want to recover it. If you're not worried about that, then no problem. And what does it do? It denatures proteins, which is incredibly effective at killing things. Destroys their membranes, both the cell membrane proper and the outer membrane of gram-negative. And of course, the DNA is going to be denatured as well. So basically everything is destroyed. Now we've got another approach using water vapor that doesn't implement using pressure, right? So this stops short of being fully capable of sterilizing in most cases, but it's called tyndallization. And what it involves is devising some sort of a chamber within which you can have free-flowing steam pass over items you want to sterilize. And notice it's in quotes because it's probably not going to achieve that, but it'll get close. You're trying to de-germ, you're trying to decontaminate some item. It could be something that you would use, say, in a laboratory as an implement there. Some foods can actually be, decontaminated this way to prevent them from spoiling too fast. So this is an approach that can be implemented there. What you do is you put the item into a container. It could be as simple as some sort of a box, maybe. And if you've got a way to get some water boiling inside of that, take a hot plate and put it in there and put a beaker of water in it and turn that on and get that boiling, that steam's going to be produced and it's going to more or less just waft over and pass over and around your object. And you do that for, the better part of an hour, something like that. And then you go ahead and remove the heat and let things come back to normal. So what happened was that heat, which is 100 degrees centigrade, that water vapor, that steam, is at 100 degrees centigrade. And, it more or less carries the heat to whatever object you're tyndallizing. And it kills all of the active cells, the vegetative cells. Now, of course, there are probably going to be some endospore-forming organisms there. So what will happen during this cooling period, which lasts for almost a day, um, is the endospores will, some of them become, uh, regenerated. They'll go back through this process of germination again, and you'll get active cells. Well, once that happens, you can go ahead and hit it with some steam again. And that'll kill off those vegetative cells. So see what you're doing is you're knocking back the number of endospores each time, and the number of vegetative cells that can come from those is limited. So you steam for an hour, you cool for a day. You steam for an hour, you cool for a day. You steam for an hour and you cool for a day. And so that typically is the cycle, 3 days worth of that. And each time the number of organisms that are present are going to decline. And this is something you could do if you didn't have access to an autoclave. If you were in a situation where it's kind of like survival mode or something like that, or you're just limited in funds, you can get things close to being sterile, but it'll never get all the way there. You can't guarantee that. So this is technically a form of disinfection, even though people sometimes say it's sterilization. It's probably going to fall short of that, and you can't guarantee it. Boiling water is not a sterilant. So when people take things like baby bottles and pacifiers and whatever else that you might want to try to remove microbes from, boiling water can't be said to sterilize. And it could get you close, but spore forming pathogens, Bacillus, the Clostridium, and other species that are like that, the gram-positive rods, they're not going to be wiped out. So generally, when you apply this, if you've got, say you're in a town where the water supply has been suspected to be contaminated with some inputs of water from elsewhere, some groundwater seeped in, I don't know, something that potentially could lead to infection. Sometimes towns will say, use the water, but boil it, and then you can use it. And 30 minutes at 100 degrees centigrade is adequate for getting rid of the non-spore forming pathogens. So this is disinfection. And then we got pasteurization, which is another application of heat. So this was developed by Louis Pasteur back in the 19th century. And he applied it first with wine. But other things were soon discovered that could be pasteurized as well. This basically helps to limit the abundance of things that could make you sick. If you were to, say, drink the milk that's not been pasteurized, just drink it straight from the cow. That might taste real good. Some people develop a taste for that if they keep cows or goats or whatever. But they run a very serious risk of acquiring pathogens that can make them very sick. All right. So if you know the people who did this kind of work, who are dairy farmers and others, they'll tell you, that it's better to pasteurize and avoid the likelihood of getting sick. And also, of course, you want to prolong the shelf life of your milk or whatever it is that you pasteurize. So what this does is it kills a lot of the vegetative forms. It doesn't kill endospores, but it kills a lot of the vegetative active forms that could be pathogenic or that could cause spoilage. And historically, after Pasteur, the temperature between 63 and 66 degrees centigrade was applied for 30 minutes. You take your milk, a large volume of it, and you heat it up to that extent. You can then go ahead and bottle it. And you'll have to refrigerate it. It can't go into your pantry at room temperature. You'll have to refrigerate that. Now, even after it's refrigerated, those organisms may continue to flourish, and at some point, you'll have surpassed an expiration date, and the food will be no good, then it will have spoiled. So now 30 minutes, that's a lot of time. And if you've got thousands and thousands of gallons of milk to pasteurize, that can be a little bit inefficient. So what they've discovered is they can elevate that temperature up to about 72 degrees centigrade. Again, not boil. Method, and that will work effectively as well. But what you're trying to do is it's sort of a trade-off. It's a the kind of balancing act between killing microbes and literally cooking the the food that you're trying to pasteurize. If you cook it and you take it too far, you're going to alter its texture, its flavor, everything about it, and it's probably not even going to be something that people will want to purchase or will want to use. How to keep that food flavor and value, and that's what pasteurization enables us to do. And then dry heat. Here they show this is an incinerator. You can use this in a laboratory to flame inoculating loops. You don't want to turn on gas if you don't have gas. Some labs don't. Can't use a Bunsen burner, so this would be a good alternative, and it works quite well. You see the little metal coils in there. It's kind of like in a toaster oven. A lot of resistance, I guess, in that metal. I don't know how they work, but it heats up. That will be enough to kill, in this case, possible contaminants. So it's being used here in aseptic technique. All right, so use higher temperatures, and you're achieving an extreme utilization of this. You're achieving incineration, which turns things to ash. If you're going short of that, you will coagulate proteins. You could denature them as well, and that of course is going to be effective too. And then cold is another application of temperature. All right, and cold is considered something that doesn't kill. Right, so it's not cidal, it's not germicidal or bactericidal, but it is static. Its microbe is static. Stasis is the word for when things have reached a sort of standstill, right? So microbistatic agents bring microbes to a standstill. They bring them to stasis. So that they're neither, it doesn't kill them outright. They don't necessarily die because of this, but they don't reproduce, and so their numbers don't increase. So this is a way to kind of flatten the curve, if you will, if you were looking at population growth of microbes, and you then go ahead and hit them with really cold temperatures. If I were to take some bacteria growing in an incubator out and place them in a refrigerator, I would stop the growth of the microbes. I wouldn't kill them, but I'd stop the growth right at whatever level. So that's why we put things in refrigerators, keep them for longer. Media, cultures, and food, all of that can be kept in refrigerators. So this isn't killing. This isn't disinfection. This isn't antisepsis. This is simply, actually, let's use the right word here. Let's just go ahead and write it out. Microbistasis. That's what we're achieving with this. All right, and then we have desiccation. So this is nothing novel. This has been implemented for centuries upon centuries by people to preserve foods, to keep them for longer. So like our own, Native American Indian tribes, the Plains Indians in particular, would kill literally tons of buffalo at a certain point in the year prior to the onset of winter, and then they would dry all that meat. Other cultures would catch a bunch of salmon and dry all that salmon. So they're making jerky. They're making things like what's called pemmican and other kinds of dried foods, even drying fruits and things like that. People in the Appalachians for centuries would grow surplus of green beans, and before they acquired mason jars and things to can them, they dried them. They strung them on strings and they hung them from the rafters and they dried them out. And then you could, take those later, like a year later, take them down and put them in boiling water and throw in a chunk of ham, and you got some nice beans to eat. So, but that's implementing desiccation, and like using cold, this only inhibits bacteria. It doesn't kill, um, uh, it doesn't sterilize or doesn't make something antiseptic, but instead it just holds the organisms at one particular level so that they no longer grow beyond that point. One way to implement desiccation, by the way, and the reason desiccation works is the removal of water. You remove water, you remove the medium within which biochemical reactions can occur, right? So all of the reactions in the Krebs cycle and the electron transport chain and things, those don't work anymore. The organisms can't function then. That's why it works. And one method of desiccation is to do that under very cold temperatures. So you get the combined effect of desiccation plus the colder temperatures. You get freeze drying, and the technical term for that is lyophilization, and that's also a preservative. Now, the organisms that live in lyophilized material can still function if you give them the right conditions later, some water and some, optimal temperatures, they'll come back to life. So while they're lyophilized, while they're freeze-dried, while they're desiccated, in many cases with prokaryotes, they're essentially just held in a suspended state of animation. So if you're a biology professor and you have to get microbes to use in your labs, one thing you can do is buy from supply houses like Carolina Biological Supply and Fisher Scientific and others. You can buy freeze-dried specimens. So you can get freeze-dried things, Pseudomonas aeruginosa and freeze-dried Streptococcus pneumoniae, and all kinds of things are available that way now. Just rehydrate them and put them in an incubator. Presto, you've got the organisms that you want to use for your labs. All right, so which of the following accomplishes microbicidal effects? Or as it says here, microbiocidal effects. Which one kills? All right. Well, freezing doesn't, but these three here actually all can kill. Now, which one can sterilize? Of all of these, it's incineration. This can be, it is a sterilant. Freezing and desiccation, these are merely microbistatic. And then boiling and pasteurization, these are going to essentially disinfect inanimate things. We usually don't use it that way. We don't think of these as disinfecting approaches, but they stop short of being sterilants. They'll kill, but they won't kill everything. So now I'm going to stop there with that one. And I'm going to go ahead and do a lecture here soon on chemical techniques. So these are physical things. We'll get into chemical things, and then actually there's some more physical things. I apologize for that. There's some more physical stuff coming. Anyway, I'm going to stop it there because I think that's adequate coverage of things for now. And then I'll put up another lecture here shortly.